when I started teaching this class like 6 years ago, um, I always went through and I talked about all the different cells and all the different parts of the specific immune system in one day. And like at the end of that day, people were freaking out. Like they were like totally blank face. Like I'm never going to understand this. This is impossible. And then the next day I would come in and do like the overview of the specific immune system and how all the cells interact with one another. And they'd be like, oh, well, that's actually not that tough. Why didn't you just teach us that first? And so after like 3 years of that happening, I was like, you know what? Maybe I'll just teach the overview first. So that's what we're going to do. We're going to go through the overview of everything. So we're not going to worry too much about what each individual cell is or what it looks like or anything like that. Just sort of how they interact with one another. And then tomorrow we'll go over what the individual cells look like and how they function and stuff like that. Okay? So first of all, for the specific immune system, we're going to divide it into two different categories. One category is for if the antigen actually gets into one of your cells, and one of them is if it's just in your body but not in a cell. Okay, so remember we got all this extracellular space in our body. So if it's in any of those fluids, what we call interstitial fluid, or in blood, it's going to be handled by one part of the immune system, and if it's in the actual cells, it'll be handled by a totally different sort of branch of the immune system. Okay, so the first one we're going to look at is called the humoral immune response. And the humoral immune response is going to be referring to body fluid. We call body fluid the humor of the body. And it comes from a really old, silly theory that the Greeks used to have about how you got sick. They thought that there were these 4 different humors in the body. They called them phlegm and yellow bile and black bile and blood. And then that any sickness that you had was caused by an imbalance of those things, right? So that's why they would like bleed people, like do bloodletting, and they would like try to extract black bile if you were depressed. They thought that like depression was caused by too much black bile, and so they would have certain other ones are caused by too much phlegm, so they would have you like constantly spit to get better. It was not super good, but this kind of hung around in the point where we still call body fluid the humor, even though we know that there's a lot more than 4 different types of it, and that it's not the thing that makes you sick, right? You're not depressed because you've got too much black bile in your body, I'm pretty sure. But anyways, the humoral immune response refers to the antigens actually in body fluid. So the response to antigens in body fluid. Okay, so we're going to do like a little flowchart type of thing here, where we're going to start at antigen being in the body fluid. So antigen in body fluid. Okay, and if an antigen gets into body fluid, it will be detected by a very specific type of cell that's called a naive B cell. So a naive B cell binds to the antigen. Okay, now let's talk a little bit about naive B cells. Naive B cells are these types of cells that have antibodies that are bound to them. They've got antibodies attached to them. Okay, an antibody is something that attaches to a certain type of antigen. Right? So essentially it's got some protein chains on it, and on the 2 protein chains, on the tips of the chains, you've got these things that are called antigen receptors, and those antigen receptors are going to be able to bind to certain antigens. Now, the very cool thing about it is that there is a antigen receptor for every single protein on the entire planet. There's an antigen receptor for every single possible protein. Okay? Every single one that can be made, there's an antigen receptor for it, which seems impossible, but really when you consider the fact that every protein is made out of the same 20 amino acids, it's not that big of a deal, right? You just have different combinations of amino acids that bind to each of those receptors, okay? So when these naive B cells are developing in your bone marrow, one of the very important things that happens is they get filtered out, okay? So as a B cell starts to develop, Here's the new B cell. It's got some antigen receptors on it. Your body cross-checks it against all of your own cells. So it says, hey, will you respond to this lung cell? No. This pancreatic cell? No. This heart cell? No. And if it goes through all your different cell types and it never responds to any of them, that's great, because that cell will be able to respond to some antigen that is not going to respond to anything in your body, right? So in other words, this B cell is not going to recognize your lungs and tell you that your lungs are a disease, right? But it might be able to recognize some other antigen that you don't have in your body and tell you that that is a disease, right? If it goes through and it creates a B cell that then cross-checks against all the cells in your body, here's a heart cell, a lung cell, pancreatic cell, and it goes ding, 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 I interact with pancreatic cells, then that B cell is destroyed, and that's not part of the B cells that are in your body, okay? So they go through a filtration system at the very beginning to make sure that you are not going to respond to your own cells with your immune response. Does that make sense? Okay. So, interestingly enough, because a lot of these cells, or because there's so many different unique combinations, there's something like 10 billion different unique combinations of antibody receptors that you can have, or antigen receptors rather, that there's not that many of each cell type. So, for instance, like if you get a new strain of flu that you've never had in your life, there's a good chance there's only a handful of cells in your body, like 5 or 6 cells, that can even recognize that that's a pathogen. Right? So very small number of cells that can recognize it. And so sometimes it takes a little while between step 1 here and step 2. Okay? So step 1, the antigen is in your body fluid. And then it might take a week before one of these B cells actually comes across this antigen and can attach to it. And at that point, you're pretty sick, because that virus has had a chance to spread throughout your entire body, and you're feeling the symptoms already. Okay? So, naive B cell binds to the antigen. Now, naive B cells, being naive, are not always super certain about what they have done or if they've done something correctly. Okay, so when it is a naive B cell, it attaches to an antigen, and it turns into something called an active B cell. Okay, so the B cell activates. Another word for an active B cell is a plasma cell. Okay, but again, it's still not sure that it's doing the right thing. Okay, and so in order to get sure that it's doing the right thing, it's going to check. It's going to present whatever antigen it's attached to on this little complex that it has called the major histocompatibility complex 2. Okay, so it's going to take that antigen. It's going to say like, hey, here's what I found. I think it's bad. Can somebody check this out for me and make sure that I did the right thing? Okay, so it presents, the B cell presents the antigen on MHC2. Okay, major histocompatibility complex 2. There's specialized cells that go around called helper T cells, and they're going to just check to make sure that the B cells are doing what they're supposed to be doing. They're sort of like the supervisors of the humoral immune response. So a helper T cell confirms the pathogen. And if that happens, the helper T cell will release these things called cytokines. So cytokines are released. Okay, and if the cytokines get released, that's going to allow this next step to go on, which is this B cell is going to divide. Okay, and when it does that, it can only divide if there are cytokines present. So we could say it requires cytokines. Okay, that B cell is going to divide, and it divides into 2 different types of cells. It divides into a cell called a memory B cell, and another cell called an effector B cell. Okay? So the memory B cells, you know how I said that you only had a couple of naive B cells that could bind to those antigens, and so it might take like a week or something before your body even realizes that it's sick, that it's got an antigen in it. Okay, so now it knows that that's an antigen that's present in your environment. You know, it knows that now flu, you know, the H1N1 flu or whatever is something that is possible to get in your environment, and so it wants to be on the lookout for that happening again. So instead of just having a couple of naive B cells that can recognize that flu virus, it's going to create a whole bunch of memory cells that now are going to be constantly looking for that. And these memory B cells are going to take up residence in your lymph nodes, and they're going to be constantly checking to make sure that you don't get infected with the same thing again. Okay? But this hasn't dealt with the problem yet. You're still sick. You still have an infection. That's where these effector B cells are going to come in, and the effector B cells are going to produce antibodies. Okay? And when they produce antibodies, that is going to be the thing that actually fights the infection. Okay? So some antibodies are effective at actually disabling the pathogens themselves, so they don't need any other steps after that. But mostly what antibodies do is they mark cells for destruction. Okay, so if it's a pathogen that is a larger pathogen that the antibody can't destroy on its own, it just marks it with a whole bunch of these antibodies and says like, hey, you know, this needs to get taken care of, and then those phagocytes will go around and destroy them because they're marked now to say like, hey, this is a bad thing, we need to destroy this. Okay, so let's talk about how this relates to your life. Okay, let's say that you get the flu vaccine this year. Okay. So you get a flu vaccine, and that vaccine has something in it called an attenuated virus, and that attenuated virus is basically a virus that is made less effective. Okay, and so you get it injected into your body humor, into your blood, right? And a naive B cell will eventually get to it and say like, hey, this is a weird thing that's not supposed to be here. Okay, it might take a week before that happens. So if you were infected with the actual flu would have plenty of time to then infect your cells and make you sick in that period of time. Okay, but you didn't get infected with the actual flu. You got infected with an attenuated flu, which is not really going to cause many symptoms. It's not really going to be able to enter your cells very rapidly, and therefore the naive B cells can take their time, and you're not really going to get very sick from it. Okay, they're going to go through this entire process here, and they're going to create memory B cells. Okay, that means when you get the actual flu, then as long as these memory B cells will also recognize the actual flu, the one that you get, you know, from you know, somebody sneezing or touching a doorknob or something like that, right? As long as these memory B cells are the ones that can actually still recognize it, those memory B cells are going to recognize it so fast that they are going to deal with it probably before you start showing any symptoms at all. Okay? And so that's how your body then responds to something that you've been infected with the second time, is that your memory B cells recognize it before it can actually cause symptoms because they're killing it off before it can get to any large number of viral particles. Okay? If, though, they, at the CDC and the WHO, if they chose incorrectly, right, and they chose the wrong flu vaccine or the wrong flu strain for any given year, then your body creates memory B cells, but those memory B cells are not effective at recognizing whatever the current strain is that's going around for the flu, and therefore having these memory B cells is not helpful. It doesn't help us to combat it in any way. But there are certain viruses that don't really mutate all that much, like chickenpox and stuff, and so it's very easy for us to give a vaccine for that, make your body produce memory B cells, and then you never get chickenpox. Okay? Before there was a vaccine for it, everybody just got chickenpox as kids. Your parents would go out and they'd have like chickenpox parties, right? And they'd be like, hey, my kid has chickenpox. You want to get your kid chickenpox this week? And they're like, I don't have much going on, sure. And so you'd all go and you'd play, and then all the kids would get chickenpox, and then you get these memory B cells in your body, and then that meant that the next time you got infected with the chickenpox virus, that you had all these memory B cells, and that would then stop you from getting sick later on. Yeah.
What do they produce again to fight the infection?
The antibodies are the thing that are actually fighting the infection, but those are produced by the effector B cells.
The memory B cells.
The memory B cells act like naive B cells. So the memory B cells do this exact same thing again. Memory B cells will bind to an antigen. They'll activate. They'll turn into, or they'll present it on MHC1. Then they will, hopefully the T cell will release cytokines. They'll divide, and they'll divide into effector B cells. So like the memory B cells are essentially just naive B cells, but in much higher quantities than they were initially. You know, you might have 4 or 5 naive B cells of each type, but once you get an infection, you'll have thousands and thousands of memory B cells that are waiting to find an infection. Forever.
That's why when you get like chickenpox, you're like, okay, you won't get it again.
Correct.
Right.
Yep.
Same thing with mono.
Same thing with mono. Yep. Yep. All right. Questions on this. What happens if a B cell screws up? What if a B cell latches on to a pancreatic cell or something like that and says, "Hey, I've got a bad thing here," and the T cell comes along and goes, "That's not an antigen. That's a pancreatic cell. We need that thing." What does the T cell do? Kills it. Kills the B cell off, and therefore that B cell will then not be able to respond to another pancreatic cell because it's dead and it doesn't get the chance to divide. There are, in rare cases, instances where B cells incorrectly attach to self proteins and they get the go-ahead from T cells, or there's maybe cytokines around from another T cell, and then it gets to divide even though it's attaching to a self protein, and then that causes what's known as an autoimmune disorder, where your immune system starts attacking your own cells, and based on the type of autoimmune disorder that you have, it has to be dealt with in different ways. We'll talk about that more tomorrow. Questions on humoral immune response? All right, let's talk about the other type. The other type that is when the antigens actually get into the cell, and that's called the cell-mediated immune response. So this is the response to antigens that have entered cells. Okay? So, step 1, antigen enters a cell. Okay? So let's say this is a virus, and we know what a virus does. A virus is going to hijack the machinery of the cell, and it's going to make that cell produce whatever viral proteins it needs to replicate, right? Yes? Okay. So at any given point, all of your cells are getting checked up on constantly. They're being micromanaged to say like, hey, what proteins are you making right now? Okay, that's fine. What proteins are you making right now? All right, I guess that's fine, right? And at any given point, the cell has to prove that it's making the correct proteins. And it does that by presenting it on something called MHC1. And every nucleated cell in your body has MHC1. That means every cell except for red blood cells. Okay? So we can say every cell, every body cell or nucleated body cell must present translational products on MHC1. Okay, when I say translational products, I mean proteins. The proteins that are being made by translation in the cell. Okay? So, if a cell is producing proteins that are viral proteins, then a cytotoxic T cell, which is the type of cell that's going to be involved here, cytotoxic T cell looks for non-self proteins. So any protein that is not supposed to be produced by the body is going to be recognized by a cytotoxic T cell. Okay? And then that cytotoxic T cell, if there is a non-self protein being produced, is going to destroy that cell. Okay? So the cytotoxic T cell destroys the cell. destroy infected cells. Okay? If it gets to do that, if it gets to destroy a cell, it's going to divide. It's going to divide into two types of cells. Memory T cells. That'll be there for the next time we get an infection. And effector T cells that are going to deal with this infection, this current infection. Okay, so these effector T cells here are the ones that are going to deal with the current infection. They're going to kill off the rest of the cells that then have been infected. Okay? Yeah?
Why do we get the common cold so often?
We'll talk about it in just... actually, we'll talk about it tomorrow. Tomorrow is the thing, is when we'll go over that. Okay? So, the T cells, though, are like not that smart. They're going to rely on the cells being honest. Okay, so this is like, imagine there's a house in your neighborhood where they're like cooking meth, right? This would be like the cops coming to the door and knocking on the door and be like, "Hey, what are you doing in there?" And the house is like, "Cooking meth," and the cops are like, "Oh, well, you're not allowed to do that." And then the cops go in and then arrest them. But most of the time, anybody who's even a little bit smart is not going to tell the cops that they're cooking meth when they knock on the door, right? So that's actually going to happen a lot in this case, where instead of the nucleated cells presenting the viral particles on MHC1, a lot of viruses will actually disable MHC1 as one of the very first things that they do. And so in that case, the cytotoxic T cell comes along, knocks on the door, and says, "Hey, what are you doing in there?" And there's no answer. And they're like, "Nah, they're not home. We'll just come back later, I guess." Right? And that's not effective. It's not a good way of killing off cells. And so there has to be another way to go about this. And there is. And that is in the event that the virus disables MHC1. So some pathogens disable MHC1. They are handled by a totally different type of cell that's called an NK cell or a natural killer cell. You can say that natural killer cells destroy any cell without a functioning MHC1. Okay, so for whatever reason the MHC1 isn't working, it could be because of a pathogen, could be because that cell isn't producing any proteins at all because it's just an old cell, or it could be because that cell is a cancerous cell that doesn't bother to present on MHC1. The NK cell, its default state is to destroy it. Okay, in other words, you have to prove that you're doing something good in order for an NK cell to not kill you. Okay, in the case of a cytotoxic T cell, you have to prove that you're doing something bad in order for a cytotoxic T cell to kill you. But NK cells, you got to prove that you're doing something good. You're doing something worthwhile. Okay? So these are good because they are going to go around and they're going to help us to combat the infections of the little bit smarter viruses, and they're also going to combat cancers and things like that. But they're bad because they kind of have an itchy trigger finger. They're going to go around and they're going to just sort of start killing cells. Any cell that's sort of in between producing proteins that may be inactive for a short period of time has a chance of being killed by a natural killer cell. So you don't want to have too many of these because they're gunslingers. But you don't want to have too little of them because they serve a purpose, right? You've got to have some of those, little shoot from the hip detectives. But you don't want too many of them because then there's no justice at all, right? So whenever an NK cell does its job and it destroys a cell, it actually is going to up the activity of the rest of the NK cells around it, right, by releasing something called interferon, right? Interferon is something that's going to essentially tell cells that there's been an infected cell and that they should be on higher alert, and that also that the NK cells should be more active in looking for cells that are infected, okay? And all these things combined are going to be able to protect you not only from viruses, but also from protists and fungi and animal pathogens and things like that, but also from cancerous cells. So cancerous cells, like I said, oftentimes won't present on MHC1 because they're just dividing too fast. And so the NK cells are really your first line of defense against cancer cells, and they're really the thing actually that does the vast majority of defense against cancer, because at all times, at any given point, we've got cells that are becoming cancerous in our bodies. And those cells are then taken care of by NK cells before they get out of hand. Anybody have questions over any the immune system stuff or the overview? Yeah.
Is this specific to humans only?
Mammals, we can say. Also birds. Yeah.
Are red blood cells not nucleated?
Red blood cells are non-nucleated. Yeah, they do not have a nucleus. The hemoglobin is too large, and the red blood cells are too small. And so there's just no room for a nucleus, so they don't have one.
Okay, so that's why there's the humoral immune response for the blood.
Yep.